In this part of the lecture, I want to talk about some different categorizations of wood. These are important both in terms of biology and physiology because of the structure function relationships associated with some of these categorizations, but these are also important because they end up having practical implications in terms of, for instance, properties of wood. So the first categorization that I want to talk about is early wood and late wood. So early wood or spring wood is just xylem cells that are formed early in the growing season. These cells tend to be larger, have thinner cell walls, and they're less dense than late wood or summer wood. The initiation of early wood tends to coincide with the beginning of primary growth in the spring. Late wood or summer wood, in contrast, is basically the opposite. Its initiation tends to happen later in the growing season and coincides with the slowing of shoot growth. Sometimes it's associated with lower soil moisture. And the late wood cells tend to be sometimes smaller, have thicker cell walls, and a smaller lumen or hole in the middle. So this is an example here of early wood and late wood in a gymnosperm. So all of these cells are tracheids, and the tree is growing in this direction. So the vascular cambium is out here to the left somewhere. So you can see these cells, these early wood cells, are relatively large. They have a large lumen or opening and relatively thin cell wall compared to these late wood cells, which are smaller, have a smaller lumen and a thicker cell wall. That transition from early wood to late wood is what gives us a growth ring in temperate trees. So when we look at a cross section of a temperate tree and see alternating light and dark bands, generally what we're seeing there is alternating bands of early wood and late wood. So you can see in this cross section of this pine, those alternating light and dark bands just coincide with alternating bands of early wood and late wood, in this case, tracheids. Those thicker cell wall tracheids and the smaller cell sizes tends to show up more dark in the larger cross section of that wood. So that's what makes those growth bands apparent, or the rings apparent. Now, you can have false growth rings caused by production of late wood in the middle of a growing season. So growth rings are not always an entirely reliable indication of, for instance, a year's worth of growth. And those patterns of growth in terms of cell size vary between gymnosperms and angiosperms. So for instance, this shows patterns of early wood and late wood in an angiosperm, in this case, aspen. Populus, this is big tooth aspen. And what you can see here is that early wood and late wood is not quite as severe, but the early wood cells, in this case, the vessels, are larger and more numerous in the early wood than in the late wood. So this, from here to here, is a growth ring in this aspen wood. So a growth ring in a cross section of aspen, in this case, may not be as apparent as it was in that pine cross section, but it's still associated with some changes in this case, vessel size associated with early wood and late wood growth. So as I mentioned earlier, that transition from early wood to late wood is often associated with changes in height growth or primary growth. So this shows, for instance, the days to height growth cessation. So when height growth stops, and its association with transition to late wood formation in loblolly pine in this case. So those two are related to each other. The phenology of height growth tends to be correlated with the phenology of late wood formation, in this case in loblolly pine. So that's early wood and late wood, or spring wood and summer wood. Another important categorization of wood is juvenile wood versus mature wood. Juvenile wood is wood that is produced within the stem when the stem is within the crown of the tree. So basically, the crown of the tree influences the activity of the vascular cambium and produces cells with juvenile wood characteristics. And we'll talk about those characteristics in a minute. But basically, let's think about wood that's formed within the crown of the tree. So let's imagine we have a relatively young tree here. In young trees, the crown tends to extend all the way to the ground. So the wood, the xylem cells that are formed during that period of time are all categorized as juvenile wood. Now, as the tree begins to grow in height, especially when it's growing in a forest, the lower branches of the crown dies, and you're left with a branch-free zone on the stem of the tree. And in that branch-free zone, the wood that's produced by the vascular cambium, or the xylem that's produced by the vascular cambium, will produce cells that are a little bit different because they're not under the influence of the crown. And that wood has mature wood characteristics. We'll talk about that again in a minute. What that means is that in a stem, there is a core of wood in the center of the stem that was produced when the crown was there, and that core of wood has juvenile wood characteristics. So if this beaver cuts down this tree, this lower log will have a core of juvenile wood with juvenile wood characteristics, and then a layer of wood on the outside that has mature wood characteristics. This ends up being important because juvenile wood characteristics end up having less desirable commercial characteristics. So juvenile wood tends to have shorter cells, so shorter tracheids or vessel elements or fibers, and that negatively impacts the strength or thickness of wood in that juvenile wood core, whereas the mature wood tends to have longer cells that are stiffer. The amount of late wood produced also differs. So juvenile wood doesn't, the rings in juvenile wood doesn't have much late wood in it. Most of it is early wood. So those rings won't have a very thick late wood ring. And that negatively impacts the density of wood in the juvenile wood core. So density is derived mainly from that late wood in the rings because it's denser. So the juvenile wood core tends to be less dense and not as strong. And then in addition, the microfibril angle in juvenile wood is greater. It has a bigger departure from vertical. You remember when we talked about that, less vertical microfibril angle means that wood is less dense. So that juvenile wood core, which was formed when the vascular cambium was basically close to the crown or within the crown of the tree, that juvenile wood core has a number of cellular characteristics that make that wood less desirable because its cell length, the amount of late wood, and the microfibril angle all end up negatively impacting the mechanical characteristics of wood that, for instance, might be cut out of that juvenile wood core. So some silvicultural decisions with regard to density of planting and the rate at which the crown recesses, for instance, are to some extent made based on how much of a juvenile wood core will be there when those stems are harvested. So that juvenile wood core is important because physiologically its cells produce wood that doesn't have very desirable juvenile wood characteristics. So here's an example that shows us, in this case, Gartner, who was a really important wood products researcher, Barb Gartner, she was at Oregon State. She sampled tracheid length in wood relative to its height above ground. And you can see this height above the ground, this portion was the part that's within the crown of the tree. And you can see the tracheids are much longer in the part of the stem that's not within the live crown, whereas the part of the stem that's within the live crown tends to have much shorter tracheid length. So that reinforces what I was telling you in terms of the cell length. So that's a less desirable characteristic of that juvenile wood core. Another important characteristic of wood is sapwood and heartwood. And we talked about this a little bit in a previous lecture. So sapwood is the physiologically active portion of the xylem. Sapwood contains vessel elements or tracheids that are transporting water, and also contains race cells that are alive or physiologically active. Whereas in contrast, heartwood is the portion of the xylem that's no longer physiologically active, doesn't transport water, and doesn't have any living cells in it. So the process of transformation of a physiologically active sapwood xylem towards physiologically inactive heartwood, that process of senescence of those living cells results in the formation of lots of chemical compounds like tannins, terpenoids, or terpenes, and flavonoids that are deposited in those cells that result in changes in characteristics of that part of the stem that may result in a characteristic color or odor. So remember me mentioning before that woods with a characteristic color or odor like black walnut or eastern red cedar are due to that transition to heartwood and the deposition of those terpenes, tannins, or flavonoids. They are what give that heartwood that distinctive color or odor or resistance to decay. So keep in mind that the heartwood at some point was sapwood, but that sapwood to heartwood transition is basically a senescence process that only occurs as stems age. So the outer layers of a stem are that physiologically active sapwood, and the senescence of that sapwood results in heartwood in the inner part of the stem. So heartwood only occurs in older stems and takes longer to develop. So older stems develop heartwood. Another important characteristic is diffuse porous and ring porous structure. This refers to structure of xylem cells within a growth ring in angiosperm. So this is just an angiosperm characteristic. So diffuse porous species tend to have basically a uniform size of vessel across the growth ring. What this means is that the early wood to late wood vessel element size is pretty uniform. There's not much variation, in this case, in vessel size across the growth ring in a diffuse porous species. So those cells don't vary much across the growth ring. So cells like species like poplar, birch, or basswood are examples of diffuse porous woods. This characteristic can be really important for identifying wood. When you look at a cross section of wood under magnification, when you see that those cells within a growth ring don't vary much, that tells you that's a diffuse porous species. Whereas a ring porous species, on the other hand, has a very big variation in vessel size from early wood to late wood. So here, in this ring porous species, the early wood has very large vessel elements and then a rapid and abrupt transition to very small, you can't even see them here, very small vessels in the late wood. That's really characteristic of oaks and hickories, for instance. Those species are ring porous species. And if you look at a growth ring under magnification, they have a very abrupt transition in vessel element size from early wood to late wood. And that's a diagnostic characteristic of those species that can be really important for instance, identifying wood from their visual by looking at them. You can tell whether they're ring porous or not, just from that early wood to late wood transition. There also are some physiological characteristics we might talk about later.